Hi everyone and welcome to my talk. My name's Adam DeLeeuw and I'm from the University of Guelph, and today I'm going to talk to you about how organisms are moving in nature. Specifically, I'm looking for phenotypically directed movement behavior, and I'll get into what that means in a second. But first I want to start off by just thanking everybody who was involved in my project, including my advisor, Baron Robinson, all my lab mates and colleagues, Keith especially, who helped me out in the field and allowed me to catch all those fish, all the volunteers that helped with the project, Ted Morwick for funding the program, of course the University of Guelph, Bob Gaucher for his cabin, and all you guys listening at home. So a large part of what ecologists try to do is study the processes that influence the distribution and abundance of animals in nature, and movement is a huge part of that. To convince you that that's the case, let's take a look at the classic population growth equation, where population growth equals the number of individuals in a population, plus the births, minus the deaths, plus the immigrants, and minus the emigrants. Movement, which is the immigrants coming in and the emigrants going out, is half the change in this equation, and has led ecologists to try to understand why and how animals are moving among the landscape. Understanding this is also imperative in managing populations of game species, helping inform conservation initiatives, and understanding the evolution of populations. So animals that move and breed in new locations are carrying their genetic material, and they're influencing how that genetic material is distributed in the environment, influencing who we see in the next generation. Which leads me into dispersal. Now, dispersal is the movement of an individual from a single site to another site where it subsequently breeds. It's typically seen as a three-step process, with emigration, the act of leaving that site, as being the first step, transfer, moving around the landscape looking for a new site, as that second step, and then the incorporation into that new site, immigration, as that third step. It's traditionally seen as either being random or directed. So random dispersal, for example, say a bird ate a seed, was randomly flying around and pooped it out, and then the seed became a tree. That'd be an example of random dispersal. Versus directed dispersal, say in salmon, where they come back to the exact same location every year to breed. So now we know that movement is not completely random in nature. Surprising, right? No, it is important to drive that point home because a lot of predictive evolutionary models assume that to be the case, so they can be misleading in their results. But some forms of directed movement include natal habitat preference, and this is where animals have an affinity to the particular habitat type in which they were born in. For example, say this caribou was born in a conifer-dominated forest. It would then prefer conifer-dominated forests going into adulthood and would probably breed there. Secondly, there's site fidelity, or philopatry, and this is where animals prefer a particular location and will breed there every year. And salmon are the perfect poster child for this, because they travel miles off the coast but always return to the exact same stream every year to breed. And there's another process called matching habitat choice, and this is where animals select habitats in which they are phenotypically matched. This is seen in a lot of camouflaging animals. And what I mean by phenotype is it can be a body form, it can be a color, or any functional trait that improves its performance in its habitat. And it's actually a relatively new concept with a lot of theoretical literature explaining where it's likely to occur, but not a ton of empirical support for this. But what the theory tells us has some interesting evolutionary implications. Now, when it comes to natal habitat preference and site fidelity, generally, these populations are specializing to their habitats through classic natural selection. This is local adaptation. This is the differential survival of individuals in habitats over evolutionary time. So for example, say we have orange fish and blue fish in an orange habitat. If the blue fish don't do too well in an orange habitat, they die off, and then eventually, over evolutionary time, orange fish will specialize to that habitat. Matching habitat choice, on the other hand, is a process in which phenotypes are sorting themselves. So in this case, the blue fish are moving out of that population. The orange fish are coming in, resulting in faster specialization. So the main question I'm asking in my study is, can I find evidence of matching habitat choice behavior in nature? And according to theory, matching habitat choice is predicted to occur in systems with a low cost of dispersal, high phenotypic trade-offs, and lots of phenotypic variability, which led me to the pumpkin seed sunfish populations of Ashby Lake, Ontario, that exhibit something called a resource polymorphism. Now, a resource polymorphism is a phenotype-environment correlation where functional traits, such as jaw size in this case, influences your feeding performance in that habitat. And in Ashby Lake, littoral fish, which occupy the shoreline of the lake, feed mostly on snails and have a much larger jaw size to compensate for that. On the contrary, you have the pelagic fish that occupy the open water, that feed mostly on zooplankton, have a smaller jaw size. In addition to these differences, we see a high phenotypic variability on the bottom of the slide here. You see that there's a lot of overlap between the littoral and the pelagic fish. And there are also identified trade-offs in the lab, so that larger jaws gives you performance benefits when feeding on snails, but is not as beneficial when feeding on zooplankton. So what I did is a mark transplant recapture experiment where I tagged 2,000 of these fish with PIT tags, which are electronic tags with a unique ID code, and exposed them to one of 3 possible treatments. The first treatment was the at-home treatment where I took it, I tagged it, and then I transplanted it back to the site where I caught it. The second treatment is the same habitat treatment, so I took it out of that capture site, tagged it, then transplanted it to a different site of the same habitat type. And the third treatment was the alternate habitat treatment, where I took it, I tagged it, and I transplanted it to a different habitat type. I then tried to recapture it later that summer and record where it went. So given that movement is a 3-stage process, the effects of phenotype could be operating on multiple stages or just one. Although due to the nature of mark recapture, I only really have information on where an individual is recaptured. And from that I can only infer which habitats it left and where it ended up, which caused me to ignore the transfer stage of dispersal. But as I mentioned before, emigration is that decision to stay or leave a site. In this case, the release site. And depending on treatment, each form of directed movement makes contrasting predictions for that decision. Natal habitat preference suggests that it's the habitat type that's important. So an individual part of the home or the same habitat treatment would be predicted to stay, and only in the third treatment, the alternate habitat treatment, where it would leave the release site. Site fidelity suggests that the site is important, and so predicts staying in the home treatment, but leaving in the same habitat or the alternate habitat treatment. Matching habitat choice makes no difference in predictions based on treatment, but rather the phenotypic match to its release site. So it would predict leaving if it is not phenotypically matched, but staying if it is phenotypically matched. So to effectively test for the effects of phenotype on leaving or habitat selection behavior, I needed a measure of how well matched a phenotype was to its habitat. And to do that, I took those original phenotype distributions that you saw earlier. And here we have phenotype on the x-axis and capture habitat on the y. You'll notice that pelagic fish have smaller jaws, littoral fish have larger jaws. If you fit a predictive line through that, that gives you a probability of occurring in a particular habitat with a given phenotype. And if you were to take the distance from the point to the line, that represents the difference from actual to predicted habitat and is actually a measure of mismatch. And I'll get into that. So these green individuals have a very similar predicted habitat to its actual habitat. So the distance between that point and the line is small. And are considered matched to their habitat. These red individuals have a very different predicted habitat given their phenotype, and so that distance between the point and the line is large and are considered mismatched. So the length of this line can then be interpreted as the intensity of mismatched a fish is to its particular habitat. So after developing this value that measures how well matched an individual is to its habitat, I can now effectively test for matching habitat choice behavior on emigration, which is that decision to stay or leave their release site. So I modeled leaving, which is yes or no, as a function of the directed movement hypotheses I identified earlier. Matching habitat choice is represented by that mismatch value, and natal habitat preference and site fidelity are theoretical models I developed based on their predictions. I then compared the fit of the models using a log likelihood test, which basically identifies whether some model represents the data better than some reference model, which is in this case the matching habitat choice model. So what we see is that mismatch has some significance in predicting leaving behavior, but it's very much outshined by the effects of site fidelity and natal habitat preference. And when I included mismatch to both of those models, they didn't explain any significant amount of variation. And after comparing the fit of all the models, it turns out that site fidelity without mismatch was the best predictor of leaving behavior. And when I graph those results, so here we see the probability of leaving with an increasing mismatch, it doesn't seem that it has too much of an effect. It's about a 6% increase, which is very meh. So with immigration, I can ask, did the fish switch habitats from its release site? And I can set up similar predictions like in emigration. Although in this case, both natal habitat preference and site fidelity predict that you stay in the same habitat as your release when you're part of the home and the same habitat treatment, but you switch habitats when transplanted to the alternate habitat type. Matching habitat choice, on the other hand, makes the prediction of switching habitats if you're phenotypically mismatched, but staying in the same habitat type if you are phenotypically matched. To model immigration, I had to eliminate the individuals who stayed at the release sites because in order to immigrate, you must emigrate. So I asked, did they switch habitats from the release site? And predicted that based off of the different movement hypotheses. In this case, phenotypic mismatch was a strong predictor in habitat switching, but site fidelity was still better. The best fitting model in this case included both site fidelity and mismatch, indicating that both processes are strongly influencing habitat switching behavior. And here we see that with an increasing phenotypic mismatch, you have about a 21% increase in the probability of switching habitats, which suggests that the pumpkin seed sunfish in Ashby Lake are exhibiting matching habitat choice behavior. And this is really cool because not only is this one of the few cases that have shown this behavior in the field, it provides an opportunity to study the effects of matching habitat choice on recently diverging populations. So what exactly does this mean? Well, firstly, my findings suggest that matching habitat choice does exist in nature, and we can add one more piece of empirical data to the puzzle of matching habitat choice. Secondly, we know that matching habitat choice operates differently on different stages of dispersal, implying here that it's more important in new habitat selection, but not as important in emigration. In other words, it helps animals choose a new location, but not leave their old one. And as far as I know, this is the first time matching habitat choice has been tested on multiple stages of dispersal. Thirdly, it's present in a diverging population, meaning that matching habitat choice here could be part of the reason that we see two different ecotypes of sunfish. And now we may observe whether the evolutionary predictions of matching habitat choice hold true, and that's exactly what's next. So that's the end of my talk. If you have any questions about the project, feel free to email me or message me. I attached my email and my LinkedIn. But anyway, thanks so much for listening. Have a great day.